Hi, everyone. I'm excited to be giving a talk today on some work that we've been looking at explaining and validating explainable AI methods using amyloid-PET classification as the task. I'm a PhD student at UCL, as James mentioned. I have no disclosures. So this work is motivated by the need to use explainability for addressing trust and bias, and which is really important for the wide-scale adoption of AI. And of course, there are legal implications of using AI in a range of settings, especially healthcare. So we need confidence in the features that we are using. And I really like this example, which isn't healthcare setting, but demonstrates the difficulty and the challenge when using black boxes for prediction. In this case, you get the right prediction that there is a horse in the image, but you can see that the model was actually looking at a little label that was in the image text to inform its predictions, and we don't want this when we're using AI for high-risk applications like healthcare. So to motivate this work, I was looking at the number of papers that have been investigating and applying AI over recent years, and you can see that there's been a rapid trend. But most of these journals are computer science journals, and there is a real gap in the healthcare setting. So last year, we performed a systematic review looking specifically at interpretable machine learning for dementia classification, and you can see that there were a large number of studies focusing on visual explanations that are derived from neural networks, like the convolutional neural network. And that's unsurprising given that we've seen high performance reported for deep neural networks in a range of tasks. And due to that, the most promising and most common method was class activation mapping, which I'll go into a little bit later. But what was also quite interesting was that, as you can see, there's a range of different methods that have been applied, but only 9 of the 92 papers actually looked at comparing different methods to one another. And this is important because there's not necessarily a ground truth when we're generating explanations. So how do we know that the method that we've chosen is the right one? So this motivates our study aims, which was to ask the question, how do we choose which method is best for my model, data, and/or task? So we aim to design an experiment to systematically compare these state-of-the-art explanation methods against one another in the case where we've got some idea of a ground truth. So the data that I want to present to you is called the amyloid-PET PNHS dataset. And this is a multi-site cohort study where they've focused specifically on early stages of Alzheimer's disease. The aim of this study was to evaluate the value of quantitative amyloid-PET measures. So they focused on a subsample of healthy controls primarily, but thinking about those with on the Alzheimer's disease risk probability spectrum. But you can see from the right-hand side that this covers a wide range of data sets and sites. And more specifically, the dataset is composed of both dynamic and static amyloid-PET. The images are not only in the native space, but also MR-aligned and available and registered to MNI space for voxel-wise analysis. But what we were most interested in was the presence of visual reads for actually labeling amyloid positivity, and this is what we use as our ground truth label. What's also nice about this dataset is that there's quantification available to derive SUVR maps, which highlight exactly where the amyloid trace has been bound to in the brain, and this is what we used as our ground truth. In the preliminary subset that I'm going to present today, there was 180 participants, and you can see here on the right-hand side the distribution of, on the top, centroid score, and on the bottom, the visual read classification that we used as a label. So I just want you to keep in mind that, as shown by the group arrow, some people who are positive or negative on visual read will be potentially what's classified as the gray zone in terms of their centroid score, and this indicates that the label is not necessarily clear. So for this task, I simply trained a model to perform amyloid-PET positivity classification. In this case, I chose the state-of-the-art convolutional neural network, or ResNet. I applied data augmentation to overcome the differences in spatial orientation. And what we are interested in doing is trying to understand what is going on in this middle section and understand exactly what features are being used. Hopefully, it will be the signal. I'll just briefly introduce to you the three methods that I've applied for explainability. The first being backpropagation, which is a technique that uses the gradients learned during training to generate a heat map. And you can alter this by using guided backpropagation to only focus on the positive gradients. I also implemented occlusion, which is a technique which is more intuitive but more computationally expensive, where you occlude certain patches iteratively and generate a heat map as well. And lastly, I applied GradCam, which is a very popular technique which focuses on using the convolutional layer to generate a heat map. So just to show you some of the results that we attained visually, first of all, you can see the model performance in the top of this figure, but model performance was not the goal of this study. We're more interested in the explanations. And you can see here the different heat maps across slices for the three methods. Unsurprisingly, occlusion gives us quite a noisy map because you're occluding patches of the image at a time. But you can see quite nicely that both guided backpropagation and guided GradCam are indeed highlighting where the amyloid has bound to in the brain. So this is a good thing. This means that the model's not just looking outside of the brain at some irrelevant feature. We are actually picking up the positivity. What I also looked at is the mean intensity difference between amyloid positive subjects and amyloid negative subjects based on their visual read label. So what we'd really want is the model to be able to the heat maps to be different for the two groups. And you can see here that guided GradCam gives us the biggest difference between positive and negative subjects on average. So that suggests that perhaps this method is best at providing or highlighting discriminative features. But what's more interesting about this data set is the presence of the ground truth in terms of the individual's PET image. And so unlike other studies which perform or generate group-wise explanations, we're able to actually look at an individual person's explanation and see if this is actually picking up their individual's amyloid uptake. And you can see here a comparison of both GradCam and backpropagation for a single person who is positive according to the visual read. And what's quite nice is that we kind of know where the ground truth is in this case. You can see that guided GradCam is showing agreement with the regions of high amyloid binding according to the PET image. So this confirms that our model is relying on biologically relevant signal. So in terms of future work, what's quite interesting is that we can start to ask other more quantitative questions, such as what is the optimal SUVR threshold that maximizes the agreement between my explanation and my PET image? You can also ask questions such as, can we investigate the heat maps in the cases of those gray zone subjects where the labelers aren't too sure whether somebody is positive or negative? Perhaps heat maps can give us some further insight that can sway an individual rater's prediction. So to summarize, my future work will be expanding beyond the small preliminary data set to use the full AMI-PAD data set, which consists of 1,800 subjects. Also look at quantifying these results through comparing the ground truth heat map with the regional SUVR values that will allow us to create some further metrics to compare. And then also exploring unequivocal cases, like I mentioned, or comparing the results across traces. So I'd like to finish by saying thank you to Professor James Cole, Professor Frederick Barkhoff, and all the team that helped put this AMI-PAD dataset together, as well as the Manifold Lab and my friends for giving me support throughout these years. I also just like to mention I've got a poster, which I'll also be at tomorrow afternoon if you want to talk more about explainability and a related project.
Well, thank you very much, Sophie. Very nice talk. We've got a few questions. Please feel free to put them in the app. So, quick question for you then, Sophie. Could the choice of reference region influence model training and results? So that's for quantifying the PET, I guess.
Yeah, I think it definitely could. So in this case, we use the visual read as the label, but what I haven't shown is you can generate a different map if you use the centiloid score as your label instead. And in that case, then yeah, the reference region would make a big difference because that would determine the value. But in this case, I'm just using visual reads.
Right, yeah, exactly. So a kind of a related question. do you think your results would change if you considered other tracers for amyloid?
Yeah, I plan to do that next. So, like I said, the AMI-PAD consortium does have another tracer. The preliminary subset just used one, but I think this is where it could be quite powerful to see if you get different important regions based on the tracer. So keep an eye out.
Okay, another question. Did you consider using other networks like a vision transformer or something like that for training?
Yes, if you come to my poster, I'll show you a comparison of the Vision Transformer and ResNet for a very similar task. I chose the ResNet primarily because training in 3D on a Vision Transformer is incredibly difficult, as I'm sure some people in here would know. So yeah, I used the ResNet primarily for that reason, but it's a good point.
Great, thank you very much. Yep. Well, thank you. One quick question that's just appeared in the chat. Sorry, Sophie.
No worries.
It's a good one for you. What is the difference between AI explainability and AI interpretability?
I think James knows I'd like this question. So I like to think of it as the difference between interpretability and explainability being in kind of when you do the prediction. So interpretability is often referred to as kind of the model itself. Is the model humanly understandable? So people often use it for sort of more traditional machine learning methods like logistic regression, where you can understand exactly how the output is obtained, whereas explainability is often used for like post hoc analysis or trying to kind of retrospectively understand what's going on. That's my take.
Great, thank you very much.
Thank you.